Hi there, my name is Matthew Raybould. I'm from the Oxford Protein Informatics Group at the University of Oxford. I'm here presenting the Evidence of Antibody Repertoire Functional Convergence through Public Baseline Shared Response Structures poster for the 3D-Sig COSY at ISMB. Our motivation for this work is that it's clear that antibodies with similar binding site structures and similar paratope residues can have the same function. However, existing sequence-based repertoire analysis techniques, for example clonotyping, only group functionally equivalent molecules that are from the same genetic lineage. In other words, have very, very similar global Fv sequences. But it's possible to have similar binding site structures and similar paratope residues, but be from completely different genetic backgrounds. And so here, instead of using clonality as a means of pooling together functionally equivalent sequences, we're going to use predicted structures as the way of pooling together potentially functionally equivalent sequences, and show that this gives us access to a whole new set of data that we wouldn't be able to currently identify. So in panel 1, here is our protocol for performing antibody repertoire structural profiling. And so you take a repertoire sequencing profile with VH and VL chains, and then we go through some steps to make sure that these chains are modelable by comparing all their CDRs to known templates from our database. And also we have to perform a certain degree of sequence identity clustering for computational tractability. Once we have predicted modelable VH and VL chains, we can perform a pairing analysis, which is looking at all the Fe structures that we have in SABDAB and seeing, okay, for each pairing, do we think we could create a realistic model? Once we have Fe pairings, we then use the templates that we identified along the way for each of the CDRs and for the Fe orientation, and use the pre-computed matrix to work out which of them represent distinct structures. And so we're able to essentially map a set of sequences onto a predicted set of distinct structures. And so applying this to samples of the baseline repertoire, so these are IgM naive samples of the repertoire that were obtained by Mariah Guidoni, and each of these 41 samples had over 100,000 VH and VL chains. So they're reasonably deep samples of the repertoire. I won't go into too much detail as to how we compared them to so-called random repertoires. You can see more information in our preprint. But essentially, what we found was that these baseline structures were occupying fairly discrete regions of structural space. So given what you'd expect by sampling randomly, was about 10 times more structural diversity than we actually saw with real repertoires, which is interesting. We also wanted to perform this overlap analysis to see which of these structures aren't just present in one particular individual, but were present in many different individuals. And so you can see with the graphic here, the way we did this was we took the most structurally diverse individual sample, so individual 1 here, and then we looked at all the Fes that we think are modelable in individual 2, and again comparing the templates of each of the cluster centers in individual 1 with all the templates from the Fes we think are modelable from individual 2, we were able to say which of those things do we think adopts a similar structure and which of them just adopt their own structure. The ones that adopt their own structure we would term private structures because they're not present in everyone, whereas the distinct structures that keep cropping up in every individual are termed public structures. After 2 baseline repertoires, we still had 30% of all distinct structures classified as public, which is much greater than the number of public clonotypes. And even after 10 baseline repertoires, we still had 3% of all distinct structures being classified as public, which is really exciting because the number of clonotypes at this point pretty much drops off to 0. And this implies a much greater degree of accessible functional commonality between repertoires than we currently know exists. So on panel 3 here, what we did was we built antibody model libraries from these sets of public structures. And so what we term the public baseline antibody model library is the actual homology models built from all 27,389 public structures that were seen after 10 different individuals. And given that the IgM repertoire in humans is so pluripotent, we thought maybe this could be the basis for a very general screening library that could be close to lots of different pharmacologically interesting antibodies. And so what we did was we compared the models that we obtained from our AML to the solved structures of several different approved or advanced stage therapeutics. And 11 of these had a representative within the antibody model library within 0.75 angstroms RMSD. That's the same set of CDR lengths and within 0.75 angstroms RMSD. And so this really gives us a lot of confidence that this could be a really useful tool to designing screening libraries that are able to bind to lots of different targets and not just one particular target. And of course, this is a geometric clustering, but the way to get chemical diversity back into this is to build your screening library based on all the FV sequences predicted to adopt each distinct structure. And so we have a structure which is geometrically similar to tremelimumab, but that particular one that we built the structure of may not be close chemically. But all the FVs that were predicted to adopt that structure over many different people, one of them is likely to be close to tremelimumab. But you don't have to have an implicated geometry to use this tool. What we can also do is apply it to longitudinal repertoire sequencing. And so in panel 4, we have a response repertoire analysis using our tool. So these are people who were sequenced before flu vaccination and then after flu vaccination. And what we're looking to see is how did the number of distinct structures change? What was the proportion of sharing? Did that change at all? And can we identify the structures that only occurred after vaccination and not beforehand? And so the key results from this panel is that after vaccination, we saw an increase in structural sharing, which is really encouraging because each of these people are sort of exposed to the same selection pressure of the same antigens upon vaccination. And so this implies a structural drift towards common epitopes. And also by clustering each individual's after vaccination structures directly after their before vaccination structures, we were able to obtain this pure after vaccination set. These are structures that are clearly not represented in each particular individual before vaccination, but are afterwards. And there's still 87,000 of these that are shared between all three individuals. And we believe that this could be the basis of a targeted screening library, for example, against influenza in this case, but could be applied to several other diseases as well. So in conclusion, about 3% of baseline repertoire structures were public to 10 different individuals, which is considerably greater than the number of public clonotypes and is consistent with a much greater level of baseline functional commonality than we currently have evidence for. Repertoire structural profiling can also be applied to longitudinal sequencing studies, and public AMLs already have been shown to contain antibodies that are geometrically close to several pharmacologically relevant antibodies, and we're excited by the potential applications for general or targeted screening library design.